I'm George Terry. I'm the Executive Vice President of Rad Source Technologies. Today we're going to have a big event here in Venlo, the Netherlands at Brightlabs. We actually have just concluded our experiment comparing gamma radiation to on-site photonic decontamination with the patented Quastar X-ray technology, and the results were astounding.
My name is Tommy Heijmans. I am responsible for the sales within Brightlabs. Brightlabs is located in the beautiful campus of Brightlands in Venlo in the Netherlands. We are a EU GMP certified lab. We do all sorts of analysis for the pharmaceutical market, but also for the cannabis market.
We got contacted actually by Spyro from DeWarrior, and Leonid asked us if we could set up and help with validating the Rad Source equipment. As we are completely GMP certified, this is up our alley. This is exactly the things which we do.
When Rad Source first came to us and said, we're very successful in decontamination and deactivation of pathogens in the US and Canada, we want to enter the European market. Picking partners like Brightlabs with 17025 certification and GMP is a proof that we could look at the results and put them in any context, in any boardroom, and those results are going to be validated. When we get to the problem itself of where does microbiology affect the product, it could happen virtually anywhere. It could happen at the cultivation phase, it could happen at the handling and post-harvest phase, it could happen at the pre-release phase. So that window of remediation happens almost immediately after you harvest your plant. You have the ability to affect that plant and to remediate the microbes. Where you act on it, how you act on it will depend on the various parameters you're working with. Are you working on purely of getting it tested positively and released? Are you worried about regrowth of colonies, in which case the options are different? Let's say you use a topical remediation such as ozone or cold plasma. You test a product, it passes, you release it. It doesn't mean that the mold at the core of that product has been deactivated, and if you have an average shelf life of 3 to 6 months of a product, you could have a colony regrowth that by the time a patient or user opens that package, it'll be fully moldy. You may have a stigma about irradiated cannabis. Anywhere between 60% to 80% of the total German medical market of 800,000 patients is irradiated.
I think this is indeed a revolutionary for the industry, because this is basically treatment in a box. Microbes have to be removed to a safe level, and one of the most standard ways to do that is to go to a gamma irradiation facility, which means it's in the hands of a third party. It's a dangerous material because it's radioactive. So it's basically a bunker. The radiation is exposed in an actual concrete bunker, and the cart comes by and the product is irradiated. So it's difficult. It's often far away from your facility. So you're talking about transport, about the security, about the transport, the packaging materials, and then you just have to wait. This machine is different because you can have it in your own facility. You plug it in, you need power and you need cooling, and then your cannabis goes into your own machine. So you control the timeline, you control the dose, you control if you want to repeat the process if it's not good enough, you can skip the process if your cannabis is clean enough, but you control all the parameters.
So the Quastar technology is reinvented X-ray. So X-ray was invented 100 years ago, and people have been using X-ray for imaging in hospitals, dental offices for years. What we did is we reinvented the X-ray so that it didn't have a focal point, and it didn't come out like a beam, like a flashlight, which puts a lot of photons in one area, and it's hard to get a good dose uniformity, which means it's hard to spread the photons equally through the product. So what we were able to do is we were able to create a light bulb type of dispersion of photons. We call it a photonic storm. It's almost like a photonic hurricane that goes around the product, that goes through the product, that and it's at a different wavelength that hits the DNA of the microbials and breaks it up so it's no longer viable, meaning it can no longer reproduce.
It's a very safe machine. You can stand next to it, you can put a glass of water in it and then drink it after it's done.
This is that glass that I put in there, but the water that's been in here has been through the irradiation process. Who wants to drink it? That's the most sterile water anybody's going to be drinking today.
Then there's also e-beam facilities, but also they require a standardized, centralized facility, usually in the hands of the government or some third party. So once again, it's much more difficult or impossible to do that in-house. So now you take a service that is mandated by the quality standards of the market and you bring it into your own sphere of influence. So I think absolutely that's a revolutionary change. The K-Star unit is basically a box with a irradiation source. It's an X-ray machine, so it's an electric way to make X-rays. When it opens up, you can put your canisters of cannabis inside. The machine will rotate so that every side of the cannabis is irradiated in the same way. So it's kind of averaged out. And then you basically set a time. The essence of this machine is, can you reduce the microbes and the fungi that are present on the cannabis without destroying anything else about that cannabis? So not the smell, not the color, not the cannabinoids or the terpenes. So anything we care about in cannabis should stay intact and the microbes should be gone. And this machine can do that. And that's not just speculation, that's what we have shown through a whole bunch of studies, including the one that was just finished at this research facility.
The meeting today was very good to explain and try to convince by telling, proving, seeing, feeling that gamma radiation is not a threat but can be very useful for our production. And we have to convince 45 years of toleration policy to do it in an industrial way, even though they get a better product. So for that reason, it was really useful today.
So in terms of potential to minimize the footprint, to ensure that we're able to do things under our control and on our timelines, I think that's pivotal to commercial success of an organization.
We've had the pleasure of working with the Rad Source team now for close to 2 years. Initially, they hired us to help advise them on market entry into the European cannabis industry. Irradiation has been used in blood transfusions, food, medicines for the better part of the last century. But in cannabis, having gone through a history of prohibition, innovation and new techniques are often regarded with much more skepticism. This particular experiment showed that even with the dirtiest sampled flower, which was somewhere around 2.5 million colonies, we were able to use the Rad Source device to remediate below the pharmacopoeia legal limits. I think Rad Source is truly commendable because they continue to spend money on research. This is now, I think, the 3rd or 4th trial they've done, State of California, Health Canada, now Brightlabs here in the Netherlands. So obviously there's more data that we're going to keep compiling, but initially I'm super pleased with how this went. So let's see what this technology does to the medical cannabis world, because it already revolutionized quite a few industries. So the potential is there, the innovation is here. The adoption has been overwhelmingly positive in the US, and now it's time to open those conversations here in Europe and in the broader pharmaceutical industry.